Okay, so when we're talking about fats in science, we call those lipids. And we first day of class talked about hydrophilic and hydrophobic things. Lipids are hydrophobic. Hydro, water, phobic. Essentially lipids are afraid of water. They don't want to be around water. We say they don't form true polymers. And the reason is because there's not one single monomer that makes up a lipid. Essentially, there's two monomers that I'll show you, and then they'll make up what we call the polymer. The lipids can kind of form a bunch of different kinds of polymers, kind of like carbohydrates will, but because there's not this one single monomer, it's not quite the standard pattern. And I'll show you what I mean in a second with the picture. Now, why are lipids hydrophobic? That's kind of a thinking you want to get yourself to at this point. It's because they are hydrocarbons. Hydrocarbons is a hydro, hydrogen, carbons, carbons. Hydrocarbons are those long carbon skeletons, like carbon to hydrogen bonds. And remember, carbon to hydrogen bonds are non-polar covalent bonds. Remember, non-polar, there's not a pole. That means the electrons are shared more or less equally in that bond. Okay? And that's because carbon and hydrogen have a very similar electronegativity. Okay? Anyways, yeah, we'll talk about fats. Phospholipids and steroids are two really big biological applications of these lipids that you'll want to know, and I'll show you their structures here in a bit. Cursor over here. All right, so here's your basic kind of how do you take the monomer and make the polymer for lipids. And I said it's kind of goofy because there's two monomers that are coming together when we're making the lipid. You have a glycerol kind of backbone, they call it, and then you have a dehydration synthesis, right? Remember dehydration synthesis? You're removing a water from the two molecules that come together. But what happens with your lipid here is you end up having these fatty acid tails that get attached to the glycerol skeleton. And typically you have 3 fatty acid tails that get attached. We call that a triacyl glycerol. Tri meaning 3, and glycerol referring to that molecule there. And but it doesn't have to be 3, and I'll show you some examples. Sometimes it could be 2, maybe even 1. That's being one like fatty acid tail. These. Again, that hydrocarbon tail that gets attached to the glycerol. Where the non-polar nature of lipids comes from is these fatty acid tails, right? Because you have all of those carbon to hydrogen bonds. Ester linkage, make sure you know that word. An ester linkage is referring to what's in that box there. So the carboxylic acid group here, that's at the end of the fatty acid tail, that alcohol group, the alcohol group from the carboxylic acid matches up with one of the alcohols from the glycerol, and then they form what we call an ester linkage, which is basically like when you have an alcohol and a carboxylic acid group, like I said here, coming together to form a covalent bond. So there's 3 ester linkages here for the triacyl glycerol. Questions so far? So yeah, obviously if you're going to only have like say 2 fatty acid tails, you would just do 2 dehydration synthesis reactions. Here you're doing 3 dehydration reactions because you have 3 fatty acid tails. Okay, make sure we set everything here. Yep. So no glycerol. It's a 3-carbon molecule, and it's got all those alcohol groups attached. Remember, alcohol group is that hydroxyl. Alcohol, hydroxyl, referring to the same thing. It's these OHs that are being attached on one side of the molecule. Yep. Oh, and those fatty acid tails, they can differ in length. So they're not always the same length that I showed you. Like they could be longer chains of carbon and hydrogens or shorter chains of carbon and hydrogens. And like how you change the length of those chains can change the property of that lipid. So again, fats separate from water because the fats are hydrophobic. They have all those non-polar bonds, whereas water is a polar molecule, so the waters want to clump together, the fats want to clump together. Remember in chemistry, like dissolves like. Okay, things that have non-polar bonds want to cluster on other things. that have nonpolar bonds. Things that are polar, molecules that are polar, they want to cluster on other polar molecules. Yeah. Triglycerides, another word. Probably heard of triglycerides sometimes, like a blood test. They'll test for triglycerides. Kind of a simpler way of saying triacylglycerol. It's kind of a hard word to say. Okay, let's talk about butter. So, how we can change these lipid molecules is basically you can add and remove double bonds to these tails. So, looking here at what we call a saturated fat, which you probably have heard that name before, right? Saturated fats, they have there's no double bonds. Reminder of like what the zigzag is referring to. Remember, each edge is a carbon, and then there's hydrogens that come off the carbons, and we can assume there's two hydrogens that I did there because carbon forms 4 bonds. So 2 of the bonds are being used to link up with other carbons. So then there's 2 hydrogens that can pair up with that carbon. Anyways, but you'll see no double bonds here. And so what happens is these tails can really pack together very closely. You'll see what I mean more when I show you the unsaturated fats in the next slide. But them being able to pack together really closely is what allows them to be solids at room temperature. Cool. And saturated fats are the unhealthy fats. That's something you want to make sure you make note of. Unhealthy fats. And the reason is they raise LDL and lower HDL. LDL refers to low density lipoprotein. I'll show you that word on a slide. I'll try it out. Lipoprotein is what that L, that ending L. First L, low, D is density. Basically, low density lipoprotein. Lipoproteins are proteins that carry fats through your bloodstream. Why would you need a protein to carry fats around your bloodstream? Why not just let the fats carry themselves to the bloodstream? Right. Think about what I said about hydrophobic, hydrophilic when we're talking about, yeah. Sorry.
So, because like the fats will just like clump together, because like they're hydrophobic.
Yeah, and then like that's an issue because like heart attacks, right? Like you don't want to clog up your arteries. So if exactly right. You need to have a protein to basically like ferry around those fat molecules throughout your body. Okay, so HDL stands for high density lipoprotein. If you have a lot of LDLs, what that says is basically you have a lot of fat going around in your body. And so this LDL, these clump easier in your bloodstream, essentially. Yes, it's a protein and it is hydrophilic, but because there's most of the density of that protein is not made of protein, it's mainly made of fat. So another way of saying high LDL is like high fat content and low protein. Right? Most of the molecule is made of fat, not as much made of protein. Whereas HDL is the opposite. There's lower amounts of fat and more of the density is protein, which means it's more hydrophilic, so it can move around the bloodstream easier. Okay. Some of these details, maybe a little bit too much you would need for like AP exam, but like knowing this part right here, high LDL, low HDL. In other words, saturated fats are the bad fats, people call them. Okay, but that would be why. Now, if we go to unsaturated fats are like your oils, right? So you'll hear the unsaturated fats being called the healthy fats, right? Think of like people talk about the Mediterranean diet, right? It's like the Mediterranean foods where you have a lot of like olive oil type stuff, like oils that come from plants. That typically is healthier for you because they do the opposite. Okay, so if the saturated fats raised LDL, lowered HDL, the unsaturated fats, we just flip the arrow. Now it's lower LDL and more of the high density lipoproteins that are more mostly made up of the protein, so they travel through your bloodstream easier. Okay. Now, what makes the unsaturated fats oils, and like what chemically, how are they different from the saturated fats? It's the double bonds. So This right here, only difference between the saturated fats and unsaturated is that double bond. So remember when you put the two parallel lines together like that, that now represents a double bond happening. And what happens to the molecule when you put that double bond is the molecule, that tail then bends. So imagine if you have like multiple of these molecules are trying to like pack together. If it bends out that like outward like that, you can't fit in together as well, right? Think of like I think we talked about like you're collecting a brush pile, a bunch of twigs together. It'll be like really inefficiently put together. But if you were to make sure all the twigs are like lining up neatly next to each other, you can then make it into a nice little bundle. Here, this is like the big pile of sticks where all those double bonds can't neatly line up next to each other. So on like the macroscopic level, it appears as an oil, not like an actual like solid like stick of butter. Questions so far? Okay. All right, I'll make sure I said everything. So this is what I was saying earlier about the saturated fats and the unhealthy fats. So again, they're going to raise the LDL, which is known as the unhealthy part of the lipoproteins. I never really said the word cholesterol. So basically, the LDLs, like if you have high LDLs, that's where you say you have high cholesterol levels. It kind of, the actual mechanism how LDL raises the cholesterol, it gets kind of complicated. Just know high LDL is going to raise the cholesterol, which is, you don't want high cholesterol levels in your body, in your bloodstream, because that's when blood clots happen. Okay. Whereas the unsaturated fats, it's the opposite story. Those double bonds, the exact mechanism can get kind of complicated, but basically the end story is how the unsaturated fats lower your LDL and raise your HDL, which then will help make your cholesterol levels in your body more normal. Okay. And it's worth pointing out, I mentioned that the length of those fatty acid chains can like be longer or shorter. The number and location of those double bonds, like whether they're like located here or there, that can change. Or maybe there could be another double bond there. But it's just same difference, right? They're still going to be liquids. It's just technically they don't have to be literally in that spot. Okay. Make sure I said everything here. Oh yeah, animal fats are going to be your saturated fats. That's why if you get like a McDonald's hamburger and you look at like the nutrition label, you'll see it's pretty high in saturated fats because the fat's coming from that. You know, if you're eating a burger, it's coming from a cow or something, right? Whereas if you get, when they say plant fats, think of like almonds, like olive oil, like oils that come from like plants, and as well as like oils you get from fish. Those are your unsaturated fats, which again are going to be healthier for you. Because you have to have fats, right? Why do you need fats? There's two big things you're going to need fats for. Yeah.
The density in terms of like how much energy.
Yeah, good. Yeah, they're very energy dense. That's a good point. Yep.
Insulation and energy
Uh-huh.
storage.
Yeah, insulation, energy storage. Yep. What do the fats make up? Think of the word lipid. Yeah.
The like cell membrane.
Yeah, the cell membrane, which we'll talk about here in a second. So, anyways, but that's why you got to have the fats. Right? Can't not have them. But that's why you want to get more of your fats from the unsaturated kinds. Okay, trans fats. So what can happen is, all right, say you get peanut butter. Anybody ever? My mom was a personal trainer when I was growing up, and she would buy like Laura Scudder's like peanut butter. You ever had that? And like you'd have to like you'd open the top and it's like just all watery and you have to like stir it up to make it all solid. I don't know if you've ever experienced this. And it's healthier peanut butter because it has more of the unsaturated fats. But when you have peanut butter, you want it to be more solid. So what they can do with like especially something like peanut butter is you'll see more trans fats in like the unhealthier peanut butters. The idea of trans fats is you get something like peanuts, right, where the oil is watery, right? If you want to make that peanut butter be solid, what you can do is you can make, instead of that double bond being a cis double bond, remember cis means on the same side, the hydrogens, now you make the hydrogens be trans. They now are on opposite sides of the double bond. what that does for you is, well, somebody tell me that. What would that do for you? So what? Why making that a trans bond? How does that help? Yeah, go ahead.
So like it keeps the structure in a straight line rather than the double bond that it's easier to like stack up.
Yeah, exactly. Yep. So now it's a straight line. So now that it's straight, and it can pack together easier, right? Whereas when it's like all like bent like that, you can't pack them together, and it's going to be a liquid. Okay. Now, it might be like, okay, then why do people make a big deal about trans fats? Well, there's just something about like these trans fats that are going to lead to more of the higher LDL levels, which again leads to more like blockages and then eventually coronary heart disease. So the exact mechanism, I don't know, and I don't believe it's really exactly known. It just does, right? It's just a bad thing. So you want to avoid trans fats as much as possible is basically the end of the story. But that would be kind of the biology or the chemistry, I guess you'd say, behind it. Questions here? Okay. Yeah, and so then kind of what we hinted at here a minute ago, like, well, we need fats. Well, why? A big function of fat is energy storage. You made a good point about the density. That's something that's not on the slide. I think it's a good point. Fats are very energy dense, meaning if you have like 2 grams of fat and 2 grams of protein, and you're on a desert island, or what does it even mean? You're on an island, like a abandoned island or something, and you're like, hey, you can have 2 grams of protein, 2 grams of fat. You should pick the fat because you get more energy out of that fat per gram than you would get from like the proteins or the carbs. Fat is good for insulation, right? Keeps you warm, and for protection. That's kind of an underrated thing, right? Like, I feel like especially like American culture, like we don't want fat in our bodies, but you got to keep in mind that you do need some fat for protection. And even it's not on here, but like it's been shown like fat will help sometimes store toxic chemicals. So like just in our diets or like in our environment, we're exposed to certain like chemicals that it's fertilizer or something like that. Those can be helped kind of stored in the fat. So the worry is if you just are like 0% body fat, like you look great with your shirt off, but like how are you gonna store like those toxic chemicals that are like kind of going around, that kind of thing. But anyways, but yeah, and fat is again, it's actually a pretty efficient way to store energy if you're thinking of like a bear going to hibernation, stuff like that. Okay. All right, phospholipids. So another reason we talked about why the cells need the lipids, probably the most important thing is the cell membrane, right? That cell membrane being shown right here, inside the cell, outside, or vice versa. It's made of basically a layer of fat. All of your cells have this layer of fat that we call the lipid bilayer. And looking at one of these phospholipids, notice how you have that glycerol backbone, right? But notice how there's only 2 phospholipids coming off of it. I want you to know that. The phospholipid, you have 2 of the fatty acid tails, and then why don't you have a third tail? Because the third carbon on the glycerol has a phosphate group. The choline group is not always there. So this is just like, it's only sometimes there. Bless you. Not all phospholipids have the choline group, but that phosphate is there. Now, the phosphate, because the phosphate has this negative charge on it, that makes the head of this phospholipid hydrophilic now. That is why the heads of the phospholipid point to the outside and the inside of the cell, because now those are going to be attracted to the water. And the tails, remember the tails are hydrophobic because of all the carbon to carbon and carbon to hydrogen bonds that are nonpolar. Those stick around the inside of the cell membrane to stay away from the water. Okay. Questions so far? Make sure you know those parts: the phosphate, glycerol, and then the fatty acid tails. Okay. Yeah, and then some of the phospholipids are going to have like that double bond there, and that's gonna, because you want the phospholipid, you want the lipid bilayer to still be kind of fluid. Like cell membranes, you don't want to be like completely rigid, Why? Why wouldn't you want it to be like packed together like butter? Why would you want some of the phospholipids to have that double bond? Why would you want some fluidity? Yeah.
So things can pass through and go into.
Yeah, good. It's good for like transportation, like diffusion and stuff like that. Also, just you move, right? You're fluid, right? You don't want like all of your cell membranes to be like completely rigid together. That's not a good way to build like a bigger organ that has some like movement and some flexibility. Cool. Okay, let me make sure I said everything here. What I didn't mention is that choline group that's sometimes attached at the very, very end. That gives a little extra stability to the phospholipid. Not as important, but is technically a thing. Yeah, make sure you know the hydrophilic, hydrophobic parts and where they point. Be able to like draw a phospholipid bilayer. And the phospholipid again is 2 fatty acids, not the 3 like the triglycerides. Yeah, interesting thing with the phospholipids, if you just add all those phospholipids into like water, they will naturally self-assemble into that bilayer. Like it's not something that like when cells make their like cell membrane, they're not really having to like put forth a ton of energy to make it happen. They just more or less naturally assemble. Now maybe when the cell has to put proteins into the lipid bilayer, which we'll talk about later on, that takes some energy, but really the lipid bilayer will just self-form. Because again, the lipids want to get away from the water, and the heads of the phospholipid want to be near the water, so they naturally form that structure. Okay, and then might seem kind of obvious, but the idea of the phospholipid bilayer, the idea of the cell membrane, is to create a difference between the inside of the cell and the outside of the cell. Right? You don't want the inside of the cell to be the exact same as the outside of the cell. Sometimes you might, but like. You know, if you're in some really bad environment where like your blood, like the outside, the fluid or outside all your cells is getting kind of chaotic, you don't want that to all of a sudden move instantly to inside the cell because then you just die immediately. Okay. Let's talk about steroids. So cholesterol is what you see here. Cholesterol is this. It's a lipid technically. But it has all these ring structures. So when we talked about hydrocarbons, I mentioned that they can have these long chains, or they can form these rings, called them cyclohexane rings. And so what you're going to notice about cholesterol and your steroids is they have these ring-like structures. So I don't need you to be able to draw this, just be able to recognize it. If I show you that, ask you what is that, that's not a carbohydrate. That is a steroid. So steroids, it's 4 fused rings, 1, 2, 3, 4. The exact number, not really as important until you get to like a biochemistry class where you have to be able to draw it, but just know it's got the rings on it. And then cholesterol will be an example of a steroid. Okay, so steroids like the general classification, cholesterol is an example of it. And cholesterol is actually really important in cell membranes. It kind of helps space apart the phospholipids so they're not as packed together, so the membrane is a little more fluid. And then this last bullet point, we talked about this, right? Where you don't want your cholesterol levels to be too high in your blood because then that's where you start getting the blood clots forming, which is like the buildup of fat in your blood vessels. That's called atherosclerosis. So there you go. So yeah, it's kind of a situation where you got to have cholesterol to make a lot of your hormones, but you can't have too high of levels because then you'll start getting the blood clots. So it's kind of a balance. And with our American diets, we don't really strike that balance very well, usually. So. Okay. Let me stop the recording here.